Welcome to everyone to the Lunch Learn Link program for today, and especially to our online viewers at University of Maryland, Howard University, the Department of Health in Maryland, and others. This talk today is one that I heard a while back, and I was, I'm not a radiation person, I'm not a physics person, I'm not an engineer, but this was very fascinating to me as as to the topic being a description of the technologies that we have available. And I know most of you will know that if you have a new technology, an improved technology, your science can go maybe a little bit faster, maybe in a little different direction. So I wanted to bring Zaver Bhujwalla's talk to us today for that reason. She's a member of the Sidney Kimmel Comprehensive Cancer Center and is co-director of the Cancer Molecular and Functional Imaging Program within the Cancer Center. I just learned that she's an NIH Outstanding Investigator Award recipient. So she's been tapped as one of those investigators who, due to their lifetime achievements already, they get an extended period of time to do investigations of their choosing. And so Xavier, I'm sure, will not run out of places to explore. And I noticed one of her talks was to image and to imagine. And I hope this talk does that for you today. Welcome, Zaver Bhujwalla.
Okay, so good afternoon, everyone, and thank you, Norma, for that very kind introduction and for the invitation to talk here today. So my title today is a little more ordinary. It's Molecular Imaging and Theranostics of Cancer, and it basically describes what we do. So we have this division of cancer imaging research in the Department of Radiology, and our goals are to promote the use of multimodal imaging to understand and effectively treat cancer. And we use different imaging modalities, many different imaging modalities. So we have a MR scanner, intravital microscopy, ultrasound, PET, SPECT, CT, optical imaging. And all of these imaging modalities can be found in the basement of the Miller Research Building. So it is a service center. And this allows us to use our imaging modality of choice to address our specific question in cancer. So the division itself has about 8 faculty members, all of whom have their own independent research programs doing really exciting work, and maybe Norma can invite them to describe their work in the future. What I'd like to do today is talk a little bit about the work done in my own lab. And so here, we've been investigating the tumor microenvironment for about 20 years or more. And so we are applying imaging to understand the tumor microenvironment and then to mine it for biomarkers and therapeutic targets. And more recently, we've expanded this to understand the effects of cancer on the different organs in the body, and we call this the tumor macroenvironment. And especially we're looking at this within the context of cancer-induced cachexia, which is this wasting away of the body that's induced by cancer. And then both of these areas are then providing insights into developing theranostic agents. And theranostics is this term that is used for combining diagnosis with therapy for precision medicine. So let me start off with a couple of examples about the tumor microenvironment. So cancer, as you know, it doesn't just consist of cancer cells. So there are things like fibroblasts, endothelial cells, lymphatic endothelial cells, immune cells, other stromal cells, and the extracellular matrix, which primarily comprises of collagen fibers. And all of these components are co-opted by cancer cells in their strategies to survive. So cancer is a disease of cell survival, unlike other diseases where the pathologies arise from cell death. Tumors are notorious for being able to withstand and survive anything that's thrown at them. And the tumor microenvironment frequently facilitates this survival and is co-opted by cancer cells. So in addition to these various compartments, also the abnormal vasculature that exists in tumors then results in areas of hypoxia, of acidic pH, and all of these then contribute to this aggressive phenotype that can form in tumors. Also, what this highlights is that the tumor microenvironment is a changing landscape. It's dynamic, it's got spatial and temporal heterogeneities, and this stresses the importance of using non-invasive imaging to follow these heterogeneities over time, especially if you are using therapeutic agents then to target the tumor microenvironment. So we have, for many years, been interested in hypoxia and specifically how it affects invasion and metastasis and how it alters the vasculature and the extracellular matrix. To investigate hypoxia in vivo, we've created cells that have the hypoxia response element linked to green fluorescent protein expression. So under hypoxia, HIF gets stabilized, binds to hypoxia response elements, and increases the formation of the fluorescent protein. And so here you can see that when you have cancer cells with this construct, they fluoresce under hypoxic conditions. And if you then make tumors from these cells, you can use this fluorescence to indirectly detect hypoxia in the tumors. And so we've now used this approach to combine it with our other non-invasive imaging modalities like MRI. Here, for instance, to understand the relationship between vascularization and hypoxia. So MRI, we've used this macromolecular contrast agent, which is albumin linked to gadolinium DTPA. GAD-DTPA is what is typically used in clinical studies to generate contrast in MRI images. Here we've linked it to albumin to make it a slightly larger molecule. And then we use it to quantify vascular volume and permeability in tumors. And so for many years, we've been seeing that areas of low vascular volume are typically very leaky. And we always thought that, yes, this was hypoxia in these areas driving up VEGF, which is this potent permeability factor. But now, for the first time, by combining these imaging modalities, we could actually see that these poorly vascularized regions were in fact hypoxic because they were fluorescing, as shown here, and they were also quite permeable. And of course, we then went on to make cells with VEGF overexpressed and showed that in fact this observation that regions of low vascular volume were permeable was then reversed when you made these cells secrete VEGF all the time. But what this also told us is that these regions of low vascular volume that were kind of doomed to die were the areas where larger molecular agents would leak out, and they would not leak out very easily in the well-vascularized, non-hypoxic regions. So again, it shows how imaging can provide insights into drug delivery in cancer treatment. So we also then wanted to understand how hypoxia might affect the extracellular matrix. And to visualize the extracellular matrix, we use second harmonic generation microscopy that then allows you to detect collagen 1 fibers through intrinsic contrast. So you see these collagen fibers in these tumors. These are, of course, ex vivo images. And so again, we were able to use our tumor model that fluoresces under hypoxia to then understand the relationship between collagen and hypoxia. And when we started this study, we thought that, yes, hypoxia is going to drive up inflammation. We're going to see lots of fibrosis in these hypoxic tumor regions. But to our surprise, what we found instead is that these hypoxic tumor regions, in fact, had far fewer collagen fibers, as shown here, and that it was the non-hypoxic regions that had the denser collagen fibers. And we've seen this now in many different tumor models, and recently we made cells with HIF1 and HIF2 silenced and found that when you silence HIF, in fact, again, collagen fiber goes up, again confirming indirectly what we see here. So then we thought, well, what is the relationship? What is the functional effect of these collagen fibers, and how does it mediate, say, macromolecular transport through the ECM? Not only macromolecular, but also low molecular weight transport, like, say, water. So for the macromolecular transport, again, we used albumin GAD DTPA and looked at its movement through the ECM beyond the vascular phase. So the first 30 minutes is all about the tumor vasculature delivering the agent, but then once it leaks out, it moves through the ECM. So if you monitor it over a period of time, you can come up with things like draining and pooling voxels in the tumor. And what we found is that these hypoxic tumor regions that had low collagen fibers also had very little macromolecular transport. So they were like silent zones in the tumor. And then we used diffusion imaging, which is another type of imaging, MRI imaging, that detects water movement. And again, we found that these collagen fibers were mediating water transport through the ECM. That again fits in with what other investigators have found, is that these collagen fibers act as avenues for cells, cancer cells, to track along and metastasize to distant sites. So here we found that these fibers were facilitating transport of macro and small molecules through the ECM. And again, this has impact for drug delivery because it tells us that hypoxic areas that have low collagen fibers, again, may not have too much of therapeutic agents then diffusing or being transported into these areas, again providing maybe one reason why hypoxia is associated with chemo resistance in tumors. So the other neat thing you can do with imaging, and here's an example, is you can look at temporal evolution by using reporters of different half-lives. So my earlier slides, we had the hypoxia response element linked to green fluorescent protein expression that has a half-life about 15 hours or so. But then that doesn't tell you much about what is happening in a shorter time frame. And so to understand that and to understand how hypoxia affects metastases, we also created cells which had both long-lived green fluorescent protein and short-lived luciferase. So luciferase has a half-life of about 30 minutes. So it can tell you whether hypoxia is existing right at that very moment. green fluorescent protein will tell you that hypoxia had occurred probably 15 hours before. And again, here we created prostate cells with this and started looking at how they metastasize. These prostate cells also have red fluorescent protein intrinsically in them. So here you can see in this example that the primary tumor itself has very little luciferase activity or GFP. So it's not very hypoxic. But in these metastatic areas, you see both luciferase and GFP expression, which tells us that the cells that moved and metastasized were not hypoxic to start with, but then were hypoxic at these distant sites. And so you can use imaging again to gain insight into the temporal effects of hypoxia and how it would influence metastasis. And of course, our whole goal here is to then understand the phenotypic changes that occur when you see these kinds of human images. So these are breast cancer patient images where you can see these are perfusion maps using dynamic contrast enhanced MRI. And you see this heterogeneity that is present. And so most likely this tumor is quite hypoxic because you see only the rim is vascularized. And so this would tell us that these are hypoxic regions and most likely contributing to a more aggressive, drug-resistant type of phenotype. Again, you see these heterogeneities in diffusion. Again, what we do in the benchtop then provide insight into what is happening in human cancers. So one other thing that we've become very interested in is the tumor secretome. And as I mentioned before, we are very interested in how cancers interact with the rest of the body. And the way tumors interact is, of course, they secrete things into the interstitial fluid that then get into the plasma and signal systemically in the body. So here we've created a little chamber that gets put into the mouse with tumor pieces around it that then engulf the chamber and allow us to acquire tumor interstitial fluid, as shown here. And here we've looked at cells with and without VEGF overexpression. And you can see that in aggressive MDAMB231 cells, VEGF overexpression changes a whole range of cytokines, as shown here, including an immunomodulator, GM-CSF, TSP1. In the less aggressive MCF7s, you don't see so many cytokines to start with, and then much fewer changes with VEGF overexpression. So again, this provides insights into what is happening in the tumor microenvironment, and also gives us biomarkers to mine for future imaging and therapeutic strategies. So our interest in the tumor interstitial fluid is also because of our interest in understanding how cancers affect the rest of the body. And so now I'd like to present a couple of examples on the tumor macroenvironment, especially how it causes cachexia. So cachexia is this wasting away of the body. And it is not primarily related to a person not eating food, because in many instances, a person will eat enough food and yet this wasting away occurs. And it's observed in over 80% of pancreatic cancer patients. And we wanted to use our imaging modalities to understand what was the cascade of events that occurred, metabolic changes in this syndrome. So to view cancer holistically and understand how it impacted the body. So to do this, of course, we again went back to our mouse studies. And we created a reporter for cachexia. So in cachexia, there's a lot of muscle wasting. And so we made myoblasts where the MERV promoter and MERV protein goes up under cachexia. So the MERV promoter is linked to TD tomato expression. These myoblasts also express green fluorescent protein intrinsically, so you can evaluate for loss of myoblasts in the muscle. So we took these myoblasts, put them into the mouse muscle, in mice with tumors that induced cachexia or weight loss and mice that did not induce cachexia. And we showed that with the onset of cachexia, so with early weight loss, this MERV promoter got switched on from this increase of TD tomato to GFP. So now we wanted to see what happened metabolically with the onset of cachexia. So here we looked at the tumor interstitial fluid in just normal mice. This is just subcutaneous fluid. Then the non-cachectic tumor and the cachectic tumor. And you can see that many of the amino acids went up in the tumor interstitial fluid of cachectic tumors. Glucose, glutamate, other substrates significantly decreased, and lactate, glutamate, succinate increased in the tumor interstitial fluid. So again, providing insights into what are the metabolic changes occurring with this syndrome, in which very little is known to date. Also of interest, the lipids, especially polyunsaturated fatty acids, significantly decreased. Of course, then we wanted to see what happened in the organs. And so here, we started looking at the liver and the brain. And you can see that, again, the cachectic-inducing tumor xenograft induced profound changes in the liver metabolites compared to the non-cachectic tumor. And similarly, for the first time, and this was actually a little scary for us, we saw significant changes in the brain. And we saw changes in the brain not only with the cachexia-inducing tumors, but also with the non-cachexia-inducing tumors. And of course, the cachexia-inducing tumors induced many more profound changes than the non-cachexia-inducing tumors. So this is summarized here. And what I'd like to point out is that in the brain, the neurotransmitters change. So GABA, myoinositol, N-acetyl aspartate, and of course a whole range of other metabolites changed in the brain. And similarly, a lot of these metabolites decreased in the liver, in the cachexia-inducing tumors, compared to the non-cachexia-inducing tumors. Similarly, there were changes in the muscle. But as I said before, our ultimate goal is to be able to detect early onset. And so this is an example from my colleague Richard Eaton and his group, where they're using non-invasive spectroscopy to detect things like GABA, N-acetyl aspartate, and this they do in about 2 minutes. And so what we now want to do is see if in pancreatic cancer patients we can detect changes in brain metabolites using non-invasive spectroscopy, because then this provides insights into potential metabolic interventions and metabolic supplements that can be used to slow down or reverse this syndrome. But again, I just want to highlight that even the non-cachexia-inducing tumors did change GABA, glutamine, and some other metabolites, and also change liver metabolites and muscle metabolites. And so what this tells us is that cancer is very much a systemic disease, and that like the tumor microenvironment, where cancer cells are co-opting the normal cells around the tumor, it is possible that tumors are also co-opting the organs of the body in their strategies to survive. And the changes in the brain, of course, suggest that if the control center is compromised, you wonder how it might be affecting things like response to treatment or immune responses and various other phenotypes that are frequently observed in cancer. For us, what we also want to do is turn this into theranostic strategies where we can then deliver things like small interfering RNA to change various metabolic enzymes, or to give metabolic supplements in these theranostic approaches. And so that brings me to the last area of focus of my talk, which is theranostic imaging. And theranostic imaging is this term that's been coined to describe a combination of detection and therapy. And so the whole idea is that you have some delivery carrier. It could be a liposome or a micelle or a viral vector. You load it with some therapeutic cargo. So we are very interested in delivering things like small interfering RNA, cDNA, prodrug enzymes. But you can use photosensitizers or radiation sensitizers. And you decorate it with some imaging reporter. And again, we have so many imaging capabilities. So it could be an MR reporter, PET, SPECT, ultrasound. And then you deliver it to some specific target in the tumor. So it could be endothelial cells, it could be hypoxia environments, it could be fibroblasts. And the whole idea is that you can visualize its delivery, make sure it's present in the tumor, and then switch on your treatment once you know that your cargo is in the tumor and has cleared out of normal tissue, thereby achieving a very precise delivery and minimizing damage to normal tissues, which is the real problem in cancer treatment, is the collateral damage that occurs. And so this is a schematic made by my associate Samta Kakad to show how this would work. So you would inject some carrier that has the cargo and the imaging reporter into the bloodstream. It would go and bind to the specific target of interest. It could need not necessarily be just one target. You could go after multiple targets. And then it would have an imaging reporter, so you could detect where it was. And then it would, once you knew it was at your target, of course it would deliver your cargo, which would be something that you could control the switching on and off of, and thereby just have very specific targeted treatment. So I'd just like to give a couple of examples of our work regarding this. So we went down this pathway of theranostic imaging some years back, as you see. And we started off to see if we could visualize the delivery of a prodrug enzyme. So the enzyme we chose was cytosine deaminase that converts non-toxic 5-fluorocytosine to toxic 5-fluorouracil, which is your typical chemotherapy agent. And we have some really smart chemists who made this with a polyl-lysine backbone to which this prodrug enzyme was attached, and we had both optical and MR reporters attached to it. And we could show that we could visualize the delivery of this prodrug enzyme in the tumor. We could wait until it had cleared out of normal tissue and then inject the prodrug, which was 5-fluorocytosine. And as a result, we would have 5-fluorouracil made only in the tumor, and it wouldn't be there in the rest of the body. So then we got a little more ambitious and thought, could we combine it with the delivery of small interfering RNA? And we have, for many years, been investigating choline metabolism in tumors, and the reason why is that most tumors have this elevated total choline. And this elevated total choline is because cancers overexpress choline kinase, which is this enzyme that converts free choline to phosphocholine, which is a component of this signal, and which is why you see elevated total choline in tumors. And so, again, the same principle. We had the Prodrug enzyme, we had small interfering RNA, except now we used polyethylamine, to which this small interfering RNA was electrostatically bound. And again, we had an optical reporter and MRI reporter. And we could show delivery of this to the tumor with imaging. We could see with spectroscopic imaging that total choline went down non-invasively because we were delivering choline kinase small interfering RNA. And we could see the conversion of 5-fluorocytosine to 5-fluorouracil in the tumor, and we got pretty good cell kill as shown here. Okay, so then we thought, okay, can we now target it to some antigen on the cancer cell? And so we said, let's try targeting this to prostate-specific membrane antigen, which is an antigen that gets expressed by androgen-independent, castrate-resistant prostate cancer. And of course, all of these theranostic strategies are targeted towards metastatic disease. So primary disease is most likely just get surgically removed, but when cancers metastasize, the options for successful treatment diminish. And so to target metastatic prostate cancer, we thought, can we deliver this now specifically to PSMA-expressing prostate cancer cells? And so here we teamed up with my colleague Marty Pomper and his group, who have for many years been developing small molecule imaging agents for PSMA. So we took that PSMA targeting agent and attached it to this Prodrug enzyme, polyethylamine, polyl-lysine structure. Now we have PSMA targeting agents. And we tested this in mice with had tumors with high PSMA and low PSMA expression. Here we use SPECT imaging because MRI is not the most sensitive imaging modality, and so we used indium-111. And we could show that this nanoparticle was retained significantly more in the PSMA-expressing tumor compared to the low PSMA-expressing tumor. And again, we were able to show that there was a decrease of total choline, and we could show the conversion of 5-fluorocytosine to 5-fluorouracil. So we hope that this we can move into treatments for metastatic prostate cancer in the future. And of course, small interfering RNA, you have many non-specific side effects. So we also wanted to see if we could make cells express something. So we used the same strategy, and instead of siRNA, we used cDNA. And we could show, again, more retention in the high PSMA-expressing tumor. And now the cDNA was TRAIL, which is a pro-apoptotic gene with green fluorescent protein expression. And we could show that, in fact, it was expressed in the tumor. And again, we could have the Prodrug enzyme converting 5-fluorocytosine to 5-fluorouracil. So this opens up many possibilities for molecular agent delivery to tumors and makes anything targetable. So if you want to overexpress something or silence something, these molecular strategies under image guidance can contribute towards that. So as you know, as I mentioned, we are very interested in hypoxia. So here, then we used hypoxia HRE-driven cytosine deaminase expression to make cells that are hypoxic express cytosine deaminase. So here's an example then that in hypoxic tumor regions, you see the formation of 5-fluorouracil from 5-fluorocytosine. And of course, YCD is now in clinical trials using viral vector delivery. So last few slides, again, this is an example of how our insights into the tumor microenvironment can be converted to theranostic strategies. So when you study hypoxia, or acidic pH, or environments that are very inflammatory, you're kind of automatically led to cyclooxygenase 2, which is this enzyme that mediates the inflammatory response of cells. And it acts on arachidonic acid and forms things like prostaglandins, thromboxanes. So we took our most aggressive breast cancer cell line and made it express short hairpin RNA against COX-2. And then applied our imaging modalities to understand what COX-2 was doing functionally. And this is just a slide to show how one single enzyme can affect so many different things in tumors and in cancer cells. So by downregulating COX-2, we found that it reduced invasion, as shown in this imaging system that we have to visualize the ability of cells to degrade ECM. It significantly changed metabolism. With Stephen Ann, who incidentally is in the School of Public Health, we showed that downregulating COX-2 reduced the ability of cells to exert forces on the extracellular matrix, so they became less muscular. In vivo, downregulating COX-2 significantly reduced collagen fibers, it reduced fibroblasts, it altered the metabolism as characterized in the interstitial fluid, and significantly reduced metastasis. So one single enzyme doing so many different things, and it highlights this interconnectedness that is there in cancer and why it is such a successful disease. So we were very excited about COX-2 and thought, well, let's see if we can silence COX-2 in these tumors in vivo. Of course, what we found is that when we used our nanoparticle that we've been using to silence choline kinase, it actually drove up COX-2. And this is something we saw with every single delivery particle. We used liposomes, we used dendrimers. They all drove up COX-2. And in effect, although we could bring COX-2 down because it shot up so much with the nanoparticle, we couldn't get it to under pre-treatment levels. So these are prostaglandin E2 levels, which is the byproduct of COX-2. So Zian Chen, who's a very smart chemist, then came up with a Dextran-based biodegradable, biocompatible nanoparticle, where for the first time, and this is about 5 years of work, we found that actually we could very successfully downregulate COX-2 in not only cells, but also in tumors, as shown here. And I think that for the future, I think all nanoparticles have to have this COX-2 test, because you don't want to give nanoparticles that are driving up COX-2 in your tumors, especially because of all the things we found related to COX-2 in tumors. So we are very happy with this, and we hope to move this into human applications, because Dextran is this molecule that's already being used in the clinic and is so biodegradable and biocompatible. So last couple of minutes, I want to talk about photoimmunotherapy. So photoimmunotherapy is this work done by Hisataka Kobayashi and his colleagues some time ago, where they found that if you take an antibody and you conjugate it to a near-infrared phthalocyanine dye, like IR700, it fluoresces, so you can detect where the antibody is bound. And then if you shine light for a little longer, the phthalocyanine dye absorbs the light and then emits it just in the proximity of where the antibody is bound, generates little holes in the cell membrane, and in effect just kills the cells where the antibody is bound. So again, you have very specific targeting. So we use this approach to go after CD44. And CD44 is something, it's a hyaluronan receptor that's expressed by cancer stem cells, is expressed by fibroblasts. And here, this is just proof of principle studies to show that we had a low CD44 expressing tumor and a high CD44 expressing tumor. When you shine light, this is the sort of damage that can be achieved with a single dose of antibody and a single dose of light. The limitations, of course, are that this works very well for superficial tumors because light cannot penetrate to a great extent. But with endoscopic or intraoperative approaches, this provides a real way for targeted precision medicine. And we are very interested, of course, in CD44 also because we see it expressed on fibroblasts. And here you can see this collagen fiber and these CD44 expressing fibroblasts in this human breast cancer tracking along this collagen fiber. And fibroblasts, as we found, again, are stromal cells that typically are associated with increased aggressiveness. Any intervention you do that makes cells less aggressive, you see the fibroblasts go down, and the collagen fibers, of course, go down as well. And so we really want to target cancer-associated fibroblasts, again, with our theranostic strategies. And so we're kind of now looking into doing the CD44 type approach with FAP alpha as well. And FAP alpha is, again, something expressed by cancer-associated fibroblasts. So here, again, is a similar thing that Jifu Jin has been doing, where he's had taken the FAP alpha antibody, conjugated it to IR700. You can detect the fibroblasts in tumors, and then we want to shine light on these tumors and see how reducing CAFs in tumors might affect different phenotypic characteristics. So I'd like to wind up there and just say how using imaging, molecular and functional imaging, to characterize the tumor microenvironment and the tumor macroenvironment, then can lead to these theranostic strategies that ultimately we hope will result in precision medicine in cancer. And precision medicine is something that is really urgently needed in the treatment of cancer. And of course, I'd like to acknowledge this great group of people I work with, without whom, of course, none of this would be possible. So this is our division. And these are the members who contributed to the work I've presented. So Samta Kakad, who does a lot of the collagen work and the collagen analysis. Balaji Krishnamachari, who is our molecular biologist and makes these cells with overexpression, silencing. Santosh Bharti, who does all the metabolomics work with the spectroscopy. Paul Winnard and Marie-France Penet, who've been working and making great strides in the cachexia work. Marie-France, of course, is also setting up her own program in ovarian cancer. Dmitry Artemov is my long-term colleague, professor of radiology and oncology, who's doing a lot of theranostic work in his own lab, but who also contributes a lot to our imaging work. Zihan Chen and Jifu Jin, our brilliant chemists, who make all these fancy nanoparticles. Jifu is also working on photoimmunotherapy. My long-term colleague, Marty Pomper and Sangeeta Ray, and Hisataka Kobayashi, with whom we do the photoimmunotherapy work, and of course support from our funding agencies. And I'd be happy to take questions.
Thank you so much, Dr. Bhujwalla. Questions from the audience? We have a few minutes. I think this gave you a picture of what's happening inside a tumor, very tangible mechanism and pictures of what's happening. questions about precision medicine?
Thank you very much for the wonderful presentation. My question is, what is the prospect for translating this lab-based science into actual therapy of patients, particularly in areas where the resources are maybe limited? Yeah, if we do it well here, what are the future prospects for translating these across?
So the imaging modalities we use are all translatable. So, you know, we have in radiology, of course, MR scanners, PET scanners, and so on. So with the tumor microenvironment, the insights that we get into, say, what's happening with hypoxia, as you recall, I'd shown these human breast cancers where you see this really heterogeneous perfusion telling us that there is hypoxia there. So it would allow us to interpret better what those images mean in terms of how aggressive that cancer is going to be. In terms of the other insights, so for instance with the macroenvironment and the metabolic changes, it opens up possibilities for metabolic interventions. So for instance, if we see that cachexia causes a profound drop in GABA or N-acetyl aspartate or myoinositol, these are supplements that you can buy them, but so far they have not been associated with cachexia. So if it looks like these are things that then we see when we move to our human studies characterizing brain spectra, then it opens up ways for just very easy translation into humans.
I'll ask this very naive question just because I'm in environmental health and engineering department. So you mentioned nanoparticles. Do you see a whole group of nanoparticles having influence on our everyday metabolism and interfering particularly with, an intent to treat?
Yes, this is a very loaded question. I was at this meeting last week on redox biology, and again there seem to be data coming out there that some of our treatments are possibly making cancers more aggressive. And I think what we do is we put a huge emphasis on growth, and treatments that reduce growth are successful, but those very treatments may be causing a more aggressive phenotype emerging. And so with our nanoparticles, for instance, yes, if they are driving up COX-2, then we definitely don't want to use them. And I think that as we advance in this field of making nanoparticles, it's a very young field just now. And, everybody is into this, look, we can do this, and look, we can do that. But as it matures, I think these are some of the things that will have to be addressed.
Well, just from today's lecture, it's very interesting how you've segmented out the various theories about cancer and what promotes cancer. So it was very interesting. Any last questions before pizza? Thank you again, Dr. Bhujwalla.
Thank you.